Today we are going to start the second method, how we can be identify the recombinant from non-recombinants. This approach, this method is called ingestional inactivations. Earlier we discussed how we can be select transformed cell from non-transformed cell with the help of selectable marker gene, because the unique feature of PBR32 is that a particular site, a particular gene which having the property to show the resistance against special antibiotics. Or in PBR322 there are two sites which can be shown the resistance against particular two antibiotics. One is called ampicillin resistance, another is called tetracycline. Then second approach, second method, how we can be select the recombinant cell from a non-recombinant cell. This approach is ingestional inactivations. Very simple method. In this method, how we can identify recombinant cell from non-recombinant. A particular DNA sequence is inserted into the sequence of an enzyme whose name is called beta-galactosidase. Beta-galactosidase. This is the enzyme. What will be happen in the coding sequence of this enzyme? Foreign DNA is insert. Due to insert of this foreign DNA, this coding sequence of enzyme become inactive. As a result, what will be happen? This enzyme, it can't show its function. It can't produce the color. It means such kind of bacterial colony which become colorless, because particular gene that will be not expressed. Due to no expression, they will not produce any kind of a color. So such kind of colony of bacterial cell is called colorless. You can say white. This is called white bacterial colonies. Instead of this one, another bacterial cell in which this outer foreign DNA is not inserted, they will express this gene which can be make this beta-galactoside enzyme, or this beta-galactoside enzyme, it can be work on a chromogenic substance. Chromogenic substrate. What will be happen? This chromogenic substance, it will develop a color that is called blue color. Here it is called white, here it is called blue. This is called colorless, or this is called colored. The color which is produced here is called blue. Such kind of bacterial colonies is called a non-recombinant, because here the desired piece of DNA which is called foreign DNA, that is not inserted, which is carried with the help of a plasmid. But in this case, the desired foreign DNA sequence that is inserted into the sequence of beta-galactosidase, as a result, this enzyme become inactive. Due to inactive, this chromogenic substance, it can't produce any kind of a color. It means it is called colorless. It is called white in color. So this is called ingestional inactivations. This is again the method by which we can be identify which bacterial cell become recombinant, which bacteria cell become non-recombinant. As the name indicates, non-recombinant, it means in which foreign DNA is not inserted. If this foreign DNA is not inserted, it means this enzyme will work properly. It will be produced the color. The color is called blue color. In early situation, when this foreign DNA is inserted into the coding sequence of this enzyme will be not work. As a result, it will be not work. This chromosomal substance, it will be not develop any color. So this kind is bacterial colony is called colorless. In short, we can say that it is called white in color or it is called blue. So this is again very important methods how we can make the distinction between recombinant cell as well as a non-recombinant cell. Another vector which can be used for cloning in plant. Very important topic. Vector which can be used for gene cloning in plant and animals. In plants versus animals. In plants, a natural genetic engineer is known. Natural genetic engineer. Very important questions. And this is natural genetic engineer is known as Agrobacterium tumefaciens. Very important question of 1 marks. What does the question say? Which plant vector is known as a natural genetic engineer? The answer of this question is called Agrobacterium tumefaciens. It is a natural bacteria which is found in soil, which can be work as a vector in a plants. That's why it is called natural genetic engineer. What the scientists will take? They will take it can be worked as a vector. The plasmid which is found in this Agrobacterium tumefaciens is known as TI plasmid. Again, very important questions. What is the full form of TI plasmid? T indicate करता है tumor. What does the word T indicate here? Tumor. Tumor. You can write TUMOR, TUMUR. No matter. I indicate करता है inducing. This I indicate inducing. Same plasmid. This is the full form of this TI plasmid, tumor inducing plasmid. It is behaved like as a vector in a plant, generally in diploid plant. Diploid plant. To deliver the particular piece of DNA, you can say a desired gene of interest into the diploid plant. So this is the role, this is the significance of this vector whose name is called Agrobacterium tumefaciens, which can be behaved like as a vector for gene cloning to obtain the multiple copies of that desired piece of DNA. You can say desired gene of interest. This can be used in diploid plant. The DNA which is transferred of this TI plasmid is also known as tDNA. It is also called transfer DNA. our DNA. This DNA contain desired piece of a segment of a DNA. You can say it will be contain gene of a interest. But naturally, this TI plasmid it will cause the disease in plant. That is called crown gall. Very important questions. Crown gall disease in plants. It means this is called pathogenic. This Agrobacter tumefacium it is called pathogenic because it will create a disease in plant which is called crown gall disease. So this vector which is called virulence it is called pathogenic. How it can be made non-pathogenic? That portion of this DNA. So this is called TI plasmid. This is called vir region. vir इंडिकेट करता है वायरुलेंस रीजन, पैथोजेनिक रीजन. Virulence region, pathogen. What scientists will done? They can remove. They make the cut here. They make the cut here. This region is removed. Now, this DNA, which is called tDNA, is called non-pathogenic. Non-pathogenic. This can be used as a vector in a plant. So generally question कहाँ से? One question that may be set up from here. Another question that may be set up about the full form of TI plasmid. Next, which is called tDNA. One marks question which is called dichotic plant. So these are the some vector which is used in plant to deliver the particular desired gene to make their multiple copies so that we can be obtain the unique characteristics in the desired plant. This vector which is used in diploid plant. There are so many another vector which can be used in monopod plant. Like in monopod plant in 1984, one virus which can be used, which can be behave like as a vector. That plant, one example, virus. It is also used to deliver the particular piece of a DNA, you can say desired gene in turnip. Another example, Gemini virus. Gemini virus. These are also behave like as a vector. These both are also behave like as a vector. Vector you know very well, that is also called taxi. That is also called carrier. That is also called vehicle. Vehicles. These are the some nickname of vector. Because what is the role of vector? To deliver the particular piece of DNA. You can say desired gene which can be inserted in the desired plants to obtain the desired characteristics, desired features. So these are the some examples. Gemini virus, it is used in mostly monopod plant. For example, in wheat, rice, maize. These are the example of a monopod. In generally wheat and maize, the particular vector are used to deliver the particular piece of DNA. And this virus is called Gemini virus, which is behaving as a vector. Like virus, which can be used in a turnip. So these are the some examples which are used as a vector for the gene cloning in plants. Instead of these, we can be used some examples of virus which is used in animals as a vector. In animals, the vector which is used is called, the most common is called animals. The most common is called retrovirus. Retrovirus. It is also work as a vector. In animal, you can say animal cell, या mammal, या mammalian cell. Another example is called adenovirus. SV40. Simian virus. SIMIN. Simian virus 40. These are also used in mammal and animal cells. Instead of this, one more example which is called papillomavirus virus. Papilloma virus. These are there are so many examples of virus which is used as a vector in a animal. Here you can see in mammal and mammalian cells. This retrovirus generally it is also pathogenic in nature. Pathogenic in nature means it will be cause the disease. How it will be made non-pathogenic? Very simple. By the disarming of vector. Disarming. By disarming of a vector. What is disarming? Very simple. The removal of a pathogenic part of vector, so that we can make a vector non-pathogenic nature. So this kind of property is called disarming of a vector. Very simple. The removal of harmful part of a vector, so that this vector become non-harmful, non-pathogenic. So this is called disarming. So same it can be performed in the retroviruses. The same can be performed in the TI plasmid, in which what we remove this part. This part is called harmful. This part is called region. As you can say, this is called virulence part, which is called pathogenic part. So we have to remove this part. If we don't remove this kind of vector, it will be causes the disease in plant. The disease which occur due to this TI plasmid is called crown gall disease. 1 marks question, very important. So these are the some vectors which can be used for the gene cloning in plants as well as the animals. Next thing we will discuss competent host. Transformations in host with the help of recombinant DNA. Very simple definition. Competent host. What is the meaning of this word competent? Very simple. It is the ability of bacterial cell to take more and more DNA from outside to inside, so that we can make a host cell. It is treated with so many different methods, chemical. It can be take more and more DNA from outside to inside. So this kind of property of bacterial cell, in which it can take up more DNA from outside to inside. This is called competent of host cell. Bacterial cell which take up more and more foreign DNA from outside to inside. This property is called competent. How it is performed? We know very well. the desired piece of DNA, you can say foreign gene, foreign DNA from outside to inside. So basically there are two methods. There are two methods of transfer of foreign DNA, या you can say gene of a interest. One method is called with the help of vector. Second method is called vectorless. Here, what does it indicate? A particular piece of DNA, या you can say foreign DNA, a gene of interest, it can be passed into the host cell with the help of some carrier. Some carrier is called vector. So this is called with the help of vector. There are so many another methods in which there is no need of vector. In such method, the DNA, a piece of DNA, a gene of interest, it can be directly passed into the host cell without the help of vector. That's why it is called vectorless. Here, with the help of a vector. So that's why it is called vector. So here, what will be used? We can use plasmid because they can carry the desired piece of DNA. Second one is called bacteriophage. Third one is called retrovirus. Yeah, different type of virus. Adenovirus, retrovirus, papillovirus, Gemini virus, poliovirus. So these are the methods which can be used. These method, which is called vectorless, these are again of so many types. Number one, electroporation. Second, micro injection. Third, gene gun method. Fourth, chemical mediated methods. There are so many methods which can be used. So these methods we will study in the next lecture in very detail. So these are the some methods which can be used. How we can be deliver the particular piece of DNA, and you can say gene of interest. So we will discuss this method one